Hi, and welcome to the Pharmacology of the Synapse. I'm Anna Lee, an assistant professor in pharmacology, and this week's topic is complex neuromodulation in addiction. In this video, we will talk about how all of these components come together in an example of complex neuromodulation. We will take a close look at the nicotinic receptor and how this receptor mediates addiction to nicotine, and the interplay between synapses in the reward regions of the brain in this process. First of all drugs of abuse increase dopamine levels in the nucleus accumbens, but do so through different mechanisms of action. The nucleus accumbens is an important part of the brain reward system. Some drugs, such as cocaine, increase dopamine through relatively straightforward methods, such as inhibiting dopamine reuptake, causing increased dopamine levels in the synapse. Nicotine acts on nicotinic acetylcholine receptors, which modulate neurotransmission, and thus its actions are much more complex. Let's take a look at how nicotine increases dopamine release in the nucleus accumbens. There are three synaptic connections in a simplified neural circuit that is important in nicotine addiction. The first is the dopaminergic pathway, pictured in yellow, that projects from the VTA to the nucleus accumbens. Dopamine neurons originate in the VTA and project to the nucleus accumbens, releasing dopamine in this area. These neurons are modulated by two other synaptic connections: glutamatergic afferents from the prefrontal cortex, shown in green, and GABA innervation within the VTA, shown in red. Both of these projections synapse onto dopaminergic neurons in yellow. Glutamate activation activates AMPA and NMDA receptors, which increase neuronal activity. GABA activates the GABA chloride channel, which hyperpolarizes the neuron and decreases neuronal activity. Thus, these two connections can be thought of as go and stop signals onto the dopaminergic neuron. If we magnify the VTA, we can see nicotinic acetylcholine receptors in the presynaptic location and the somatic location. These receptors on the dopamine neuron and on the presynaptic GABA neuron are alpha-4 beta-2 subtype. The receptors on the presynaptic glutamate neuron are alpha-7. So let's take a look at what happens when nicotine arrives to the system, such as when a cigarette is smoked. Acute administration of nicotine activates all of the receptor subtypes. Opening the alpha-4 beta-2 nicotinic receptors on the dopamine neuron will allow cations to enter the cell, resulting in increased neuronal firing of the neuron and increased neuronal activity. This causes more dopamine to be released in the nucleus accumbens. Activation of the alpha-7 and the alpha-4 nicotinic receptors on the glutamatergic neuron and the GABA interneuron will also increase activity in both neurons, but since one is excitatory and the other is inhibitory, they essentially cancel each other out, and the overall effect is driven by the dopamine neuron. During continued nicotine administration, the receptors begin to desensitize due to prolonged agonist presence. The alpha-4 beta-2 nicotinic receptors in this system desensitize more quickly than the alpha-7 nicotinic receptors. This will change the balance of neuromodulation on the dopamine neuron. The inhibitory GABA input in red will decrease, represented by the thinner line here, but the excitatory glutamatergic input will be maintained. This will result in a net go signal from the glutamatergic neuron onto the dopaminergic neuron. Glutamate from this projection will activate AMPA-NMDA receptors on the dopamine neuron, thus increasing neuronal activity of this neuron, resulting in more dopamine release in the nucleus accumbens. Now, after chronic nicotine exposure, the receptors undergo the phenomena of upregulation. Normally, when an agonist is chronically administered, receptors respond by downregulating their expression to reduce agonist activity. This is a pharmacodynamic response to the drug and maintains organism homeostasis. Nicotinic receptors have an interesting property of upregulating after chronic agonist administration. This upregulation is seen in vitro, in cells, in animals, and in human smokers. The fact that it is seen in vitro suggests that the mechanisms underlying upregulation do not require an intact organism and originates within the cell. The exact mechanisms mediating receptor upregulation are unclear. It was originally thought to be due to a homeostatic response to receptor desensitization, but further research has shown that nicotine itself may act as a molecular chaperone that speeds up the maturation of immature receptors in the cell and helps move the receptors to the surface. In smokers, upregulation of nicotinic receptors is reflected by increased binding of nicotinic receptor ligands. This is a figure of PET scans of the binding of an alpha-4 beta-2 radiolabeled ligand called 2FA in smokers versus non-smokers. So the control are the non-smokers and the smokers are below here. You can see that red and yellow colors show areas where more radioligand is bound, and the colors are warmer in the smokers' brains compared with the non-smokers' brains, reflecting an increase in receptor binding and receptor expression. Upregulation of receptor expression is thought to play an important role in the escalation of nicotine intake and the development of drug tolerance in smokers. Fortunately, upregulation of nicotinic receptor expression can be reversed, such that ex-smokers show similar nicotinic receptor binding levels as never smokers. In the next series of videos, we will take a look at how some smoking cessation drugs act to help people break their addiction to nicotine.